the conclusion of almost all these important information I just mentioned. So let's go back to see the slides. Okay? This is the slide showing you palisades. Underneath phase contrast microscope, which is called bacterial diphtheria, you see the club shape. Very obviously, club shape. We will do a throat culture testing in the lab. Some of you will get Corynebacterial diphtheria, not really diphtheria, Corynebacteria in your throat culture. So we'll see. Some of them get in the year before the pandemic. I didn't do this last year. You see the gram-positive generate exotoxin, inhibit protein synthesis, and the resistance to drying. Now, where are we going to get these diphtheria or this stuff? All these infectious disease, of course, in a crowded area. Is that right? That's a common sense. If nobody there, you're not going to get it. Same thing as the COVID-19. In a crowded area, if you don't have a mask, you don't keep social distance, of course you're going to get it. It's very simple. So here it tells you what it looks like. The bacteria is localized in tonsil, and they have a cell membrane right there. And the toxin goes everywhere, goes blood, goes kidney, heart, nervous system. Therefore, they have neurological symptoms, like difficult to speak, difficult swallowing, and double vision. And this is the typical symptoms. You see a very greenish, soft, and when you touch it, it's going to be bleeding, cell membrane system. And you see the kids has a very heavy swelling of the neck, just like a ball neck, because the lymph nodes is swelling. So what we should do, we need to give them antitoxin to neutralize exotoxin. And we give them antibiotics, but very important, everybody should be protected in the United States. And the protection rate is very high, more than 90%, because we have a vaccine called DTaP. This is what they should do. Okay? 2 months. When you get a birth, hepatitis, this you give to mom. Okay? What do you see? 2 months, you're gonna have a DTaP. Diphtheria, tetanus, pertussis. 4 months, you're gonna do another shot. 6 months, do another shot. 1.5 years, do another shot. 4 and 6 years, you do one more. This is called a booster. Remember, we're talking about the booster these days all the time. Is that right? Why we need this booster? For the bacteria, you know, when they into the body, you're going to activate the 2 line of the immune system. Is B cell line and T cell line, which is cell-mediated immune and the humoral immune. For most of these vaccine, like DTaP, is only going to activate the humoral cell-mediated immune. So they do not generate really strong antibody, because they are not like other things to activate humoral immune, which means the B cell. So their memorization in your body is very short. That's why you're going to keep give the booster all the time. So you see the 5 shots for DTaP to let the people get protected. And then most of them, you see pneumococcal, Hemophilia influenza, we're going to mention real quick. That's all the same. You know, influenza is 1 year. The only thing we're going to give 1 shot is measles, mumps, and the rubella, which is MMR. And remember, if you come to the United States as a foreign student, you need to get that one. Otherwise, university will not let you registration for the class. But only 1 shot. Is that right? Because that is coming from a live bacterial cell. It's a live toxin. Okay? So these are things we'll mention later on when we talk about the vaccine. We'll talk a little bit more. So just give you an idea, we do have a DTaP can protect the kids in the United States, but they need a booster all the time until 4 to 6 years old. This slide, which tells you a mechanism of the diphtheria toxin, how they work. And I mentioned it's an AB toxin. It's a binding domain. A is toxin domain, B is a transmembrane domain. So when they go inside, originally the pH is 7. But when they become in the sun, the pH is lower, become 5.0, then they release. So the B components recycled out of the cell surface to find another A coming inside, like a carrier. Because they're transferring, so that's why it's called bleeding, cause bleeding. Then the A toxin domain to prevent the elongation factor 2 for the protein synthesis. Okay, of course, they are actually prevent the nicotinic adenine dinucleotides, NAD, to combine with elongation factor 2. And this is another one which will show you more clearly how they prevent the protein synthesis. Okay? So these are all we're going to talk about. The first one, we talk about bacterial diphtheria. Second part, we're going to spending pretty much lots of time going to talk about another second group of a major bacteria, which is called meningitis. So first of all, what is meningitis? Okay, this is the information between your brain membrane and spinal cord. Now, what are the symptoms for meningitis? First of all, of course, you had a headache. Lots of you have. You have some fever. Some vomiting could be. And very sleepy. Lethargy. And another thing, scared to the light. We call it photophilia. Scared to light. And don't forget to one more thing, people doesn't realize. Tough, stiff neck. This is typical for young kids, for the kids in college. Okay. These are something we want to mention before we move on to talk about bacteria. Now, what are the bacteria will cause meningitis? We had a table there, show you the detail. But I want to tell you one thing. Those pathogens is age dependent. If you are very young, a newborn, 0 to 2 months, the bacteria caused meningitis is Streptococcus agalactiae. This is also called GBS, Group B Streptococcus. I will talk about what that means next Tuesday. Group B Streptococcus. I use STREP indication Streptococcus, okay? Otherwise it's going to be the same. Okay, 2 months to 5 year old kids. Which pathogen will cause meningitis? Type B, Hemophilia influenza. We're going to talk about this one very quick. We also call it HIV. Type B, Hemophilia influenza. Okay, how about people a little bit adults? 40 to 40 years old. The pathogen caused meningitis is Neisseria meningitis. We will talk real quick. Then how about older people? I was just barely past 40 years old. Streptococcus pneumoniae. We will talk this on next Tuesday. Okay? This is age dependent. Well, of course it's not really. You have kids who had a streptococcus pneumonia, has pneumonia symptoms. If you don't do anything, it will cause meningitis. Besides like coughing, like sneezing, crowded area in the lung area, that's possible. But typically, it's age dependent for these bacteria. So we want to talk about these one by one. So first of all, Hemophilia influenza. Okay. H-E-O, hemo, H-I-L-I-U-S, Hemophilia influenza. First of all, why this one called influenza? When they do the research a long time ago, when they first find this bacteria, they find it kind of like flu-like symptoms. They did not realize it's meningitis until very late. So they think it's maybe like just an influenza, maybe just like a flu. So they name it Hemophilia influenza. Okay? But they didn't realize it will cause more severe symptom, which is meningitis. That's why they say Hemophilia influenza at the beginning. Okay, first of all, what are the characteristics? This is gram-negative, very tiny Coccobacillus. What means Coccobacillus? Like a cocktail, but also like a logs. That's why they call it Coccobacillus. And you will see most of our bacterial infectious disease pathogen is Coccobacillus. Number two, this bacteria is a fastidious bacteria. Now what this means? Okay, clinically we have three agar we'll be using. Macanthi agar, blood agar, and the chocolate agar. Okay, what means this is a fastidious bacteria? So it's not going to grow on the Macanthi agar, not on the blood agar, only on chocolate agar. It's a fastidious bacteria, which means it's very picky. Okay, why? Because this bacteria, when they grow, they need XV factor. What is X factor? Is Hemi. What is V factor? NAD. They need both. So they did a research. Okay, they have agar. This is X factor. This is a V factor. This is XV. They're not going to grow here. They will grow just surrounded right here. Become turbidity. Okay, now some of them say we usually isolate the spinal, cerebral spinal fluid sample from the patient. Then we find that those guys sometimes grow on the V factor. Why? There is a subgenus which is called Parainfluenza. This is caused mild symptoms. Okay? It's one of the subspecies. But what we talk about, we're not going to talk about this guy. We're going to talk about this real Hemophilia influenza. Okay, so that is a very picky because they need the X and V factor, and the chocolate agar provide that. Then the second thing what happened is, when they do the clinical testing in the lab, they find that if you have a blood agar, this is a blood agar, and you grow a Staphylococcus aureus, and then you inoculate Hemophilia influenza there, what are you going to see? You see it's also grow very tiny. These we call satellite phenomenon. Okay? So these are the two things. This one I should use. I should use a very dark white, red color. I don't have it. Okay, this is a blood agar. Just want to show you it's a blood agar. So these are the two things we could do to identify Haemophilus influenza. Now, where they got these, how they got these isolation, usually coming from CSF, cerebral spinal fluid. Okay? So then, let's go back to see some of the slides. So we move on to another one. Let's go to see slides. Meningitis, we mentioned the inflammation of brain and spinal cord meninges. Now, by the way, what is the inflammation? Do you know? What's the symptoms of inflammation? You're gonna feeling a little bit of heat, a little bit of red, a little bit of swelling, and of course, it's gonna be hurt. Okay? These are the 5 major symptoms for inflammation. Okay, how we do it? We get cerebral spinal fluid, CSF, from the gram stain. Now, culturing CSF may or may not grow bacteria. It depends. Sometimes it's a fastidious bacteria. Is that right? So what are the pathogens will cause meningitis? We mentioned Streptococcus pneumoniae, Neisseria meningitis, Haemophilus influenza, because it's age dependent. Okay? How they transfer? Respiratory drops. What are the symptoms? Lethargy, confusion, vomiting, headache, and a tough neck, stiffness neck. Maybe fatal, yes, maybe recovered very soon. So we mentioned about Haemophilus influenza here. And I also want to mention, this is we call #1 childhood disease, long time ago. Before this comes out, before the type B Haemophilus influenza comes out. Okay, now why we say it's a type B? Because this bacteria also has a heavy capsid. Type B, heavy capsid. So when they do the manufacturing the vaccine, they are coming from the heavy capsid of the bacteria. So that's why it's type B, HIV, the vaccine named that. And this one comes out is after 98 years. Before that, not really helps too much. That's why curing 3 million worldwide a year. And in the United States, it's been good. It's still about like 20,000 cases every year, if you look at some of the survey results. And the vaccine is the best one to control it. They reduce case to less than 1 per 100,000 people. Okay, so that's Haemophilus influenza. This is the slides, what I'm showing you talk about is right here. So you can see, it's a fastidious bacteria. When you're using chocolate agar, it turns brownish. I mentioned it's no chocolate there, it turns brownish. Blood agar, it grows very well. Single colony. And this is the one which means in a neutral agar, they need the X and V factor. You see the X and V factor there is surrounded? X factor is hemming, V is X factor is hemming, V is NAD, nicotinic adenine dinucleotides. This one, what I mentioned, is a satellite phenomenon. So you have a Staphylococcus aureus grow, and then you inoculate Haemophilus influenza, you will see it's a tiny, teeny, like bacteria surrounding that. Because blood agar will be supplied hemming. You know that. That's why it has the blood there. And during the growing process of the Staphylococcus aureus, it will create nicotinic adenine dinucleotides, which is NAD. It's an electrocarrier. We'll talk real quick. That's why it can grow. That's called the satellite phenomenon. Okay? So this slide is very important to tell you. Hemophilus influenza is a very picky, fastidious bacteria. Okay, so this is what I have. Then we're going to move on to talk about the Neisseria meningitis. This one we will mention again later on when we talk about sexual transmitted disease, because Neisseria gonorrhea is the same thing. See, it is the same genus name. So we will talk a little bit briefly here. This is Neisseria meningitis. Okay, first of all, Neisseria meningitis. When we talk about Neisseria, Neisseria. When we talk about Neisseria, it's very easy, relatively easy to be differentiated because it is gram-negative. We call it large shape, but we still say it's bacillus, and we call it coffee bean arrangement. Looks like that. Is that a coffee bean? You all drink like instant coffees, right? You don't do those beans, coffee beans, you cut it. This looks like a coffee bean. And usually you see on the slides, there's a large chunk there. When you see slides with a large chunk like this is white blood cell. And surrounded, and in the center like a coffee bean. Okay, we need to use a coffee machine to cut it, is that right? Smash it. So very typical. It's a coffee bean arrangement. Second, Neisseria meningitis. The risk group is undergraduate. Is undergraduate students in residential hall. Very crowded area, or you call it storm. Okay. I'll tell you the story behind that. Number 3. In the United States, there are 5 toxins or serotypes were responsible for Neisseria meningitis. It's A, B, C, Y, and W 135. This bacteriological serotyping is responsible for 40% of the case in US annually. Okay, now there is a story behind this. It says there's a vaccine. Is it right on the slides? The vaccine developed very early, around the 1990s to before 2000. This vaccine called Meningitis. It covers A, C, Y, and W 135. Okay, there is a debate. Undergraduate, remember, they fit this category, 5 to 40 years old. Now, there are some people who is older, I mean, it's okay, but majority of the people in this category, okay? So there is a resident home. There is a risk group. Once the vaccine manually comes out, in the California, Connecticut, and Massachusetts, they have to have shots. Otherwise, they're not allowed to register for the class. However, there's a debate. You cover ACY 135. You don't cover B. What that means? Roughly 60% protection. Is that right? You can do the math. Roughly. So some states are debating, well, I don't want them to, even the people in those areas still mad. I don't want to give the vaccine because it's only 60% protection, 40% of the chance, barely lower than 50%. You know, 50% is like coin toss, is that right? Everything is 50%. So they don't want to get it. Now, why they cannot have the vaccine to cover the type B? Because Neisseria meningitis, very similar to the Hemophilia influenza, they also get a capsule from the bacteria to create a vaccine. We will mention there's a lots of ways to create a vaccine. Some of them use a live cell, some of them use a dead cell, some of them use a polysaccharide, some of them using a chemical synthesized or genetic synthesized. Like COVID-19, they use RNA protein, is that right? So there are different ways to do. At that time, they are using the capsule. When they isolate the B serotype, they find the capsule polysaccharides is identical to the human cell polysaccharides. So that's caused a problem. You know, if there's the same, your body will not recognize as a foreigner, will not activate the immune system. You know what's a vaccine, which means it stimulated a lower dose of the infection in your body. That's why they're going to activate your immune system. They activate the B cell or T cell. If the capsule polysaccharides of the type B and the human cell, they're identical, it's going to be not doing anything. So what they do, after the monument has been created, around like 2010, they started to get surface protein of type B, serotype. And they isolated about 600 surface protein. And they did a screening test in mice. They find 25 of them are useful. And then they from the 25 surface proteins further to do a clinical trial. And I don't know whether they're successful or not. That's all the material comes out, stopped from the page of the textbook. So around 10 years ago, they started to do type B serotype. And the latest news, around like 2017, they have the vaccine, which they claimed could be cover the type B. Okay, but then they don't talk about it anymore because we had a COVID-19, they don't care about that anymore. Okay, so just want to let you know the history, this one, the high